Okay, so as Dennis introduced me, I'm the senior analyst in Sanjay Sisodia's group at the University College of London. And today I wanted to present you a methodology I got interested in a while ago, published almost a year ago in Nature Genetics, which is actually a gene-based association method which makes use of imputed gene expression data. So the methodology can be simplified in 3 steps. So starting with a reference data set, which is genotype measured for mRNA expression, we develop prediction models for gene expression in the sense of multiple SNP prediction models with weights, the SNP and gene. Then those prediction models can be used to infer unobserved gene expression in genotype data sets somebody wants to study. And then finally, those prediction expression levels can be correlated with the phenotype in a gene-based association test. So those 3 steps graphically represented here. So we are starting from the reference transcriptome with genotype matrix of the SNPs, genotype per individual. And then in the same individual, we have the transcriptome data with the gene expression level per gene. This can also be coming from different tissues. And with that, we predict models for gene expression, like in the sense of weights for all the SNPs for every gene. And then with this matrix of weights per SNP and gene, we can go to our data set, which should be genotyped or imputed for the same SNPs, and impute the transcriptome. And then together with phenotype vector, calculate gene-based association tests. So now the question might arise how well this imputation of gene expression data does work. So the answer is good and also maybe less good. You have here an example of 4 genes which can be imputed rather well. You can see on the x-axis the predicted expression plotted to the observed expression on the y-axis with, in all cases, rather high R-square correlation scores. But then if you look genome-wide to all genes, you might see a different picture. So we see here on the y-axis again the square correlation scores between imputed expression and observed expression. And we do have, every gene is represented by such a red spot here. And we start with rather high correlation scores, almost close to 1. And this goes down up to almost 0. This is depending on this black line, which depicts the heritability of the gene expression. Heritability in this sense can be calculated in the same way as you would calculate the heritability for just traits based on the concordance of genotypes between unrelated samples. So out of this graph, you can assume or can take maybe one of the limitations of this model, that in the end, you will not be able to have a gene expression model for every of the genes in the exome. So like, for example, in the study I'm presenting, which and this also depends on the number of samples in the reference transcriptome. So the study I'm presenting used one of the largest transcriptome data available of 900 blood samples, and the number of genes with prediction model is around 9,000 only. So then to complete actually this methodology, I have to show you also a recent publication from bioRxiv this year, in which the developers of Prediscan, is actually the software called, expanded it in a way so that this methodology can also work with GWAS summary results instead of individual level data, which might not always be available. And this was especially a response actually to a competitor group which published this year in Nature Genetics similar approach, which they called transcriptome-wide association analysis or study, which more or less does the same to impute gene expression. They just use a different statistical model to do so, but they have a big advantage of with their initial published software to be able to use directly summary association statistics. And then, of course, with this new publication of the PredictXcan group, they did state that their methodology has advantages over the TVAS methodology, because the Gaussian imputation underlying TVAS is presumed to be suboptimal for gene expression trait modeling. This is an observation taken out of one of their own publications from PLOS Genetics this year. So this is then, in the end, the combined workflow options we have with both software tools. So either starting from individual level data with genotype and phenotypes, we can use PredictXcan to predict the transcriptome and then calculate gene-based association scores. And in the case GWAS or meta-analysis were performed, individual level data are not available, we can just take SNP level summary results, use metascan, and generate the same gene-based association results. And then, of course, they do show that both methods, so the producing the same results with almost perfect correlation. And you can see here the results with association results using summary statistics plotted against the summary, the association results using individual level data. With perfect correlation, as I said, and with the few outliers explained by differences in the LD calculation, which has to be done for the SNPs, which is for PredictXcan based on the individual level data set, and for Metascan, it's based on a reference data sets like the 1000 genome data sets. So then to emphasize maybe the simplicity for the user, especially of the whole methodology, for PredictXcan, the user is just to come up with genotype data which have to be imputed, and the prediction models for gene expression are provided. This is it. While for the MetaXcan software, this user has only to provide summary statistics from GWAS or meta-analysis, and provided a reference set in order to compute the structure of the SNPs, and the same prediction model as for PredictXcan is provided. Now, coming to an example of real association results, this is from the PredictXcan publication, analysis of the Wellcome Trust dataset from 2007 of type 1 diabetes. And you can see in the upper Manhattan plot the original single marker GWAS results, and in the plot below the association results per gene of the same data set. And you can see on the first side that the gene associations are consistent with single variant GWAS results, with obviously the only difference that we do have a direct candidate or several candidates for these associated loci. And then apart from that, this is maybe the main advantage of this whole methodology, which can be depicted very nicely in the case of the gene ARBB3, which is a gene which was mentioned in the well-contrast GWAS as a potential candidate gene, and then later confirmed to be indeed associated with type 1 diabetes. So this is an example of a table, the results of the Prediscan methodology, which doesn't show anything special. Here's the p-value and SNPs used as predictor for the gene expression level. But one of the most important values we have here is the direction of effect. So, which tells us what kind of gene expression level is associated with the phenotype. In the case of ARBB3, a lower expression of this gene is associated with increased risk for type 1 diabetes. And this association with a lower expression rate of this gene is supported by publication from 2010, where they could show that protective genotypes for type 1 diabetes are associated with a higher ARBB3 mRNA level. And then maybe just one last point finally, so for all who are working with gene-based association tools on combined sets of raw and common variants. This is a comparison now also for one of the well-contrast data sets, in this case rheumatoid arthritis, where they compare the QQ plot of the associations of the gene using Prediscan in green, and then using two of the other commonly used gene-based association analysis, which can incorporate raw variants and common variants, which is the second kernel association test, CUT, and maybe less known Vegas. And you can see that Prediscan tends to generate lower p-values. So then summarize the published tools, Prediscan and Metascan, are SNP aggregation approaches, which are integrating information on whatever SNP regulates the expression of a gene. Reverse causality is not a major concern because the disease status in the drug treatment does not alter the germline genomic variation, which might be, so this reverse causality might be a case in all of the studies which are directly measuring gene expression and then performing gene expression phenotype correlation. Then it's interesting to see that the identified gene association are consistent with single variant GWAS results, of course with the advantage that they allow direct biological interpretation. The developers pretend that the detected association can be generated with smaller sample sizes than compared to standard single SNP GWAS. Both methodologies are very easy to use. And of course, the main advantage, as I said, is the provided directionality of low or high gene expression. Okay, and this is it. And because I see we have a bit of time left, maybe I can show you one more aspect which can be taken out of the publication, which is the question maybe if different transcriptomes from different tissues are available, which one should somebody use for his disease? And one aspect is they investigated how expression models developed from blood samples do predict the expression levels in different tissues. And this is the observed prediction correlation plotted to the null distribution. So when the two vectors would be independent, and this departure from the null distribution is telling us that in this case, models derived from blood were able to significantly predict the gene expression level in also different tissues. So we are able to, if this stands this way, to use also gene expression levels derived from blood samples to investigate diseases like epilepsy, where we will assume that the tissue affected must be something else but blood. So, and then in the end, this is the available transcriptome data sets for both software, so which are provided to be as ready to use prediction models. So the first is coming from the Genotype Tissue Expression Project datasets, TTAX. From the last version, they uploaded to dbGaP of 44 human tissues with 70 to 301 samples per tissue. And of course, once again, depending on the number of samples per tissue, from 2,000 up to 8,000 genes per tissue with prediction model. And a second model they also provide from the depression genes and networks whole blood data set with 900 samples, the prediction models for around 9,000 genes. And they show in their recent publication from Metascan that it is of a certain value to investigate any kind of phenotype in all available tissue. Thank you for your attention. This is it.